Our next speaker is Nathaniel Safren, who will talk about the genome-wide CRISPR interference screen reveals commander genes as the modifiers of glucocerebrosidase activity and lysosomal function.
Great. So today I'm going to talk about some of the work I've been doing in Dimitri Krantz's lab. All this work, yeah. Sorry about that. We should start off by saying all of this work was a collaborative effort between myself and a fantastic postdoc in the lab, George Manakaki. This was an equal contribution effort between the two of us. It's been a really great collaboration. So George and I set out to identify genes that both positively and negatively modulate GCase activity to better understand the biology of GCase and GBA1-PD, but also to potentially uncover genes that may play a role in GBA1-PD penetrance and PD risk more broadly, and also to identify new potential therapeutic targets. To do this, we performed a pooled genome-wide CRISPR interference, or CRISPR-I screen. In CRISPR-I, a gene of interest is targeted by a catalytically dead version of Cas9, and it's fused to a transcriptional repressor, which turns down gene expression. We use this approach to target every protein-coding gene in the genome with a lentiviral library with 5 guides per gene. We performed our screen in cells with a wild-type GBA to give ourselves the maximum sensitivity to identify genes that, when knocked down, reduce GCase activity, because these are the genes that are normally essential for its function. We infected cells with our lentiviral library, and then enriched for infected cells, and then incubated the cells with the lysosomal GCase reporter, PFB-FDB. We were confident that this approach was suitable for a screen based on work that we had done with LIMP2 knockout cells. LIMP2 was brought up before. It's a necessary co-transporter with GCase, and so we view it as the optimal positive control for genetic modifier. In LIMP2 knockout cells, we see about a 90% reduction in the PFB signal. We performed our screen 3 times, each time collecting 2 fractions of cells. Those cells with the highest and lowest 30% of GCase activity. And we sent these away for sequencing to obtain guide RNA frequencies. And if a gene has an overrepresentation of guides targeting it within the low fraction, normally that gene is a negative regulator of GCase. A necessary regulator of GCase, because when it's knocked down, activity goes down. Conversely, if a gene has an overrepresentation of guides to high fraction, it normally negatively regulates GCase. So with our sequencing data, we were able to visualize the results of our screen on a volcano plot. Here, red dots are significant hits, and those to the right are those genes that, when knocked down, reduce GCase activity. And reassuringly, both GBA itself and SCARB2, the gene which encodes for LIMP2, came up as hits in the screen. Providing further construct validity for our screen, a number of other genes involved in lysosomal homeostasis came up as hits, including the transcription factor MITF, which was covered before, as well as members of the Hops complex and RAB7, which mediates the fusion of endosomes with the lysosome. But most importantly for the rest of the talk, some of the prominent hits in the screen were members of the COMD gene family. And these included the top 2 hits in the screen, COMD3, and its transcriptional read-through protein, COMD3-BMI1. We validated that loss of COMD3 reduces GCase in cells. We produced 2 independent knockout clones, and we see about a 50% reduction in PFB activity, or PFB signal, independent of the PFB uptake itself. And we were able to reproduce these findings in iPS-derived microglia, and using the lyso-F2-GBA compound in forebrain iPS-derived neurons as well. So given that we could be confident that COMD3 loss reliably reduced GCase, we next wanted to determine how this was happening. And so to investigate this, we looked at the role of commander proteins in a larger macromolecular structure called commander. In commander, the 10 COMD proteins form a heteropentameric ring that binds to CCDC22 and 93, as well as DEND10, and this constitutes the CCC complex. This further expands to the commander complex when it stably associates with the retriever complex, which mediates retromer-independent protein sorting. Commander has been ascribed a role in linking retriever cargo in the membranes of endosomes with the wash complex and the actin cytoskeleton. And in this way, it's thought to help with the transport of retriever cargo to the plasma membrane. This complex could be visualized in cells by staining for one of its constituent proteins, COMB1. And you could see these COMB1 puncta. In keeping with a previous publication in COMB3 knockout cells, we see destabilization of the commander complex, which can be observed as a dissolution of these puncta. Add back of tagged COMB3, but not the tag itself, fully restabilizes commander, and crucially for our purposes, fully restores lysosomal GCase activity on the knockout background. Beyond just seeing a deficit in GCase activity, we also see a deficit in lysosomal proteolysis using the DQ-BSA assay, which also could be restored with COMB3 add-back. So this observation prompts us to look at the abundance of proteins within the lysosome via lyso-IPs. And there we could observe a significant decrease in GCase, LIM2, LAMP2, and cathepsin B. And we were also able to see this in lyso-IP fractions collected from dopaminergic, COMB3 knockout dopaminergic neurons. Importantly, in COMB3 knockout cells, we see no shift in the expression of any of these genes, nor do we see any shift in the expression of genes within the T-Fed transcriptional network. So given that these proteins are being expressed, but they're not reaching the lysosome, we wondered if they could be misrouted. And so this was brought up briefly before, but one of the alternative routes for alternative fates for endosomal proteins that don't reach the lysosome is to be secreted as exosomes. This is a regulated process in which intraluminal vesicles fuse with the plasma membrane, and then exosomes are secreted. So to look at this, we collected exosome fractions using two different methods, ultracentrifugation and immunoisolation. And in both cases, this yielded fractions that were enriched in exosome markers, flotilin and ALEX. And in exosomes derived from COMB3 knockout cells, we detected the presence of LIMP2, and this you could not observe in exosomes derived from wild-type cells or knockout cells with COMB3 add-back. We similarly detected LAMP1 and HEXB as well in these exosome fractions, and we were able to observe this in exosomes from COMB3 knockout dopaminergic neurons. So given the impact of COMB3 loss on PD-relevant pathways, we next wondered whether loss-of-function variants in COMB3 or commander genes might play a role in PD risk. And to look at this, Bernabe Bustos, a postdoc in the lab with an extensive bioinformatics background, performed case-control gene-set burden analyses of rare predicted loss-of-function variants in the COMB ring, or genes that comprise the COMB ring, the CCC complex, and the commander complex. And Bernabe, and he did this using data from the AMP PD data set and also the UK Biobank. And in all three sets, he saw an increased burden of these variants, suggesting that these variants are modifying risk in these two independent populations. So to summarize this project, a genome-wide CRISPR iScreen nominated these COMB genes as regulators for GCase activity, and we knocked out COMB3 and found that it indeed decreased GCase activity. Upon add-back, you could restore it, and that in COMB3 knockout cells, it seems that multiple lysosomal proteins are being misrouted and aberrantly detected in exosomes. And finally, the rare predicted loss-of-function variants and commander genes are overrepresented in at least the two data sets we looked at in PD patients. So with that, I'd like to thank Georgia, who has been an incredible collaborator, and Bernabe, who performed all of the human genetics analyses, as well as Dimitri, Nicolo, and Stephen for guidance and resource support.
Thank you. Any questions? This is fascinating. Yeah.
So that's a great question. Of course, we want to see, is it actually modifying penetrance? That is like the whole point of the project in the first place. Just statistically, these are rare variants, right? So by definition, that means they can't be more than 1% in the population, and then you're also further slicing your pie with people who have to be GBA1 positive. So we just can't answer that question.
Time for one more question, maybe, if there is one. Thank you very much.